So this week we're going to be talking about chapter 24, which talks about the DNA viruses. So viruses that have DNA as their genetic material. Remember, a virus can either have DNA or RNA, but it never has both. So remember, all viruses need to have that genetic material, either the DNA or RNA, and then they also need that capsid, which is the protein that surrounds and protects the genetic material. Remember, some viruses have envelopes and some are naked. And many animal viruses will also have spikes that are used for attachment to the host cells. And the spikes is what makes a virus very specific. First, let's just review some of the main characteristics of viruses that will be important for the DNA virus chapter. And next week, we'll talk about the RNA viruses. So these rules also apply to the RNA viruses. So remember, viruses are obligate parasites. They need to get inside of a host cell in order to replicate. They can't reproduce or replicate on their own. Now, most DNA viruses, most of the ones we're going to talk about, are double-stranded. There's one exception, and that is the parvoviruses. You might have heard of parvo. It's often in animals. It's one of those things that you hear about at the vet, but it also can affect humans. And parvovirus is the only single-stranded DNA virus. Now, on the other hand, RNA viruses are mostly all single-stranded, with the exception of reoviruses. Reoviruses are double-stranded RNA. We'll talk about them next week. Remember, every different virus is very specific to its host, as well as the type of cell that it infects, which you'll see throughout this chapter. Most DNA viruses will enter into the nucleus of the cell because the nucleus is where the DNA is, and this is where DNA replication takes place. The exception to this, as you'll see, are the Poxviruses, and the Poxviruses replicate in the cytoplasm. RNA viruses, because RNA, right, is made at ribosomes, so RNA viruses mainly stay and replicate inside the cytoplasm of the host cell. Remember that many viruses are transmitted through zoonotic transmission, either through animal to human or through vectors like arthropods, like arboviruses, or viruses could be human to human, and some viruses can be fomites to human. So every virus spreads in a very specific manner. This table is very nice in organizing all of the DNA viruses that we're going to go through, and we're going to go through them in this order. So we're going to start with talking about the variola, also known as smallpox. Then we're going to go into the herpes viridae, which includes herpes simplex virus, VZV, cytomegalovirus, Epstein-Barr, and then human herpes virus 6 and 7. Then we'll talk about hepatitis. The only DNA hepatitis is hepatitis B. Hepatitis A, C, all the other hepatitis are RNA viruses, but we'll talk about them briefly in this chapter. And then we'll talk about some of the other DNA viruses. As you can see, a lot of the DNA viruses do have an effect on the skin or skeletal muscle, but they do affect other systems as well. Lastly, just to review some of the effects of viral infections. Remember, a viral infection could be acute, also known as it goes through a lytic cycle, meaning the virus will cause a rapid onset and a short duration, which could be a serious infection or could be mild, and it always depends. Or a virus could be persistent, meaning it's either lifelong or lasts for many years. If the virus is constantly reproducing throughout the course of the infection, it's a chronic infection, such as, for example, hepatitis B. And then if the virus goes through periods of outbreak and dormancy, this is known as a latent infection, such as, for example, herpes simplex virus. Don't forget, if you see that word oncogenic, it means that the virus is associated with cancer. So mostly persistent viruses, and as you'll see, some of these DNA viruses are actually oncogenic. If the virus is teratogenic, that means it can cross the placental barrier and potentially cause developmental or permanent defects to the fetus. And as you will see, there are some DNA viruses that are teratogenic, mainly in the herpes family. Diagnosing viral disease we talked about in the last chapter. A lot of times we'll use ELISA or other serological methods, such as Western blot, or they might use genetic methods, genotypic methods, such as PCR. So the DNA viruses are broken up into 7 groups, and you'll see they spell happy. So the H is herpes viruses, and then another H for hepatinovirus, that's hepatitis. The A is the adenovirus, and then there's actually there's actually 4 P's: poxvirus, polyoma, papilloma, and parvo. The way that we categorize DNA viruses, first we look at whether it's enveloped or non-enveloped. So you can see here, we go to envelope. All the enveloped DNA viruses are double-stranded DNA, and they are the poxvirus, herpes, and hepatina. And then the non-enveloped, there's either the double-stranded DNA, which is adeno, polyoma, and papilloma, and then that one single-stranded DNA group are the parvoviruses, also non-enveloped. So this is the order we're going to go through today. We're going to start with talking about the enveloped viruses, and we'll start with the pox viruses. So pox viruses, this is a family of viruses. They're enveloped double-stranded DNA. These are the only complex-shaped viruses. They actually look like very large. They almost look like a cell because they basically have 2 envelopes. So they have this extra layer on the outside. Now, what's unique about pox viruses is even though they're double-stranded DNA, they don't replicate in the nucleus. They replicate in the cytoplasm in something called factory areas. And specifically, they like to target the epidermal cells. So pox viruses can spread through skin-to-skin contact or maybe potentially respiratory route. They're also quite large, and they have the largest genome of all the viruses. So one of the examples of a pox virus is smallpox. So smallpox used to be an extremely devastating disease. As I'm sure you've read about in history books, smallpox is also known as variola. And there's actually a few different strains of variola. Variola major, this was a strain of smallpox that was highly virulent. It actually could cause toxemia, toxic shock, and intravascular coagulation. So this was the most serious form of smallpox. There was also variola minor, which was much less virulent, maybe localized to the skin. There's also something called vaccinia. Vaccinia is a very close relative to variola, and it was actually used to make the smallpox vaccine. So smallpox is actually the first and only disease to be completely eradicated in the entire world thanks to a vaccination. When smallpox was around, like I said, it would spread through inhalation or skin contact. It would cause these things called pox, which are these little pustules on the skin. They would eventually become crusty. It could lead to fever, fatigue, rash, etc. So like I mentioned, the vaccinia virus was used to make the smallpox vaccine. They actually would put this virus onto this double-pronged needle, and they would puncture the skin, and it would form this scab that left a scar. You might actually know somebody, maybe from another country, who's older, who might actually still have this scar. But we do not get this vaccine anymore because this disease is eradicated. Routine vaccination ended in 1972. The only people who may still get this vaccine are certain high-risk military personnel. And that's because there are still a couple of frozen vials of smallpox left in some labs, one in the United States and one in Russia. And there's always worry that it could turn into a bioterrorist weapon. So certain high-risk individuals may still be vaccinated, but otherwise we don't get vaccinated anymore. There are some other pox viruses that are much more prevalent still today. One of them is molluscum contagiosum. This one actually causes these small kind of pox, but they almost look kind of like warts. They are often in the genital area because this is actually sexually transmitted. It spreads through skin-to-skin contact. So in the United States, it's most commonly an STD, or it could be seen in like young children, like at a daycare, because it can be transmitted through fomites and through close touching, things like that. It mostly will stick on the legs and in the genital region. And like I said, they'll usually appear as kind of like small warts almost, but they're usually removed by either freezing or by chemicals that can remove them. So that, because although they don't really cause a lot of harm or medical problems per se, they just kind of are not aesthetically pleasing. So many people choose to have them removed. Individuals with AIDS may see these pox form on their face, and they may form more like a tumor-like growth instead of just a small pox. There are a few other types of pox viruses that humans are susceptible to. For example, monkeypox. which we don't see very often unless a human is in close contact with monkeys. Now, monkeypox could also be prevented with the smallpox vaccine, but because we don't get this vaccine anymore, humans are now more susceptible to getting monkeypox. And it produces kind of a similar disease to smallpox, only maybe a little bit less severe. So again, like pox, fever, swollen lymph nodes. Humans also could become infected with cowpox, although again, very rare. Back in the day when there used to be milkmaids, they would see cowpox maybe on the milkmaid's hands. Today, we don't really see it much from cows, but other animals like mice or cats could pick it up from a cow, and then a human could potentially come in contact with it from their cat or something like that.